In this episode, we discuss how the three-dimensional structure of our genome controls the fate of every cell. Our guest Viviana Risca at Rockefeller University is uncovering the molecular mechanisms behind chromatin organization, deciphering how DNA folds and interacts to regulate gene expression. Using innovative chemical and computational tools, her research reveals the hidden architecture of our genome and its profound role in development and disease. Today, we delve into the complex world of chromatin mapping and what it tells us about the blueprint of life.
Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello, Viviana. I'm happy to have you on the show today.
It's my pleasure to join. This is a wonderful podcast, and I'm very honored to be included in the list of really respected colleagues that have been on your podcast.
Yeah, I'm very excited to go along your scientific journey. So the first question I always like to ask to ease into the topic is, how did you become interested in biology in the first place and then in pursuing a career in science?
So when I was very young, I was actually interested in astrophysics and astronomy. And when I really fell in love with biology was in ninth grade. I had an amazing teacher named Judy Ferris who went much deeper into molecular biology than was strictly necessary for the New York State curriculum. And I think the aha moment was when she described how ribosomes work, because I had heard about DNA and DNA sequence, and I had heard about proteins, but it didn't really make sense. And so when we got that lecture about the structure-function relationship between tRNAs and the process of translation and the ribosome, effectively exchanging information from nucleic acid sequence to amino acid sequence, I was just shocked. And I remember walking out of the classroom that day just with a giant smile on my face because it finally all made sense. And I was pretty hooked after that. And so the same teacher was very active in talking to folks at institutions in New York City. And she mentioned one day that a professor at NYU, Dan Eichinger, was looking for a high school volunteer. And my hand just shot up immediately. And next thing I knew, that summer after ninth grade, I was commuting into New York City from the suburbs and working in his lab and learning how to do PCR and cloning. And after that, I worked almost every summer in a lab, and I got to learn biology that way.
So how did you then end up in the field of epigenetics? Because epigenetics, it's a big field and definitions vary, but it's different than biology and it's also different than ribosomes, right? So how did you then end up in epigenetics?
Right. Why am I not a translation researcher? The thing that really fascinated me about it was this idea of information transfer and information encoding. And during the summers in high school, I had come across this article about DNA computing, which was just kind of emerging as an idea at the time, where people were starting to try to encode logic gates and small bits of information into DNA to use it as a very efficient biological storage medium for data. And because I had been interested in physical science before I got into biology, that really appealed to me. And so I did this project one summer, sort of in my bedroom. Actually, sorry, no, it wasn't the summer. It was during the academic year. In my bedroom, trying to optimize a little PCR reaction for the purposes of a DNA computing operation. And when I presented it at a science fair, Carter Bancroft from Mount Sinai was one of the judges and invited me to work in his lab to do sort of a basic DNA computing project. And so I ended up doing that, and that ended up getting published. And I also submitted to science fairs, and I got a scholarship that allowed me to pay for a big part of college. So I got to go to Stanford. And when I got to Stanford, my goal was to study computer science and biology together because I wanted to work at this interface of information science and biological science. Um, I ended up actually being a physics major because as I was sort of trying to take different classes and talking to different people, I ended up having a meeting with Steve Chu, who was a professor at Stanford at the time. And he convinced me that physics was kind of like the liberal arts education for for the sciences, where if you study physics, you can kind of do anything. And I felt very comfortable in biology, but I didn't always feel like I was completely comfortable in physics. It was a muscle that I wanted to develop further. And so I decided I would challenge myself and be a physics major at Stanford, take mostly physics classes and the biology basic requirements for a minor, and then get as much of my biology education as possible by doing research over the summers. And so I ended up in this biophysics space, even though there was no official biophysics major. And I was interested in both information processing and structure-function relationships. So when it came time to choose labs in graduate school, I ended up going to UC Berkeley. I had a really hard time deciding. I was trying to decide between Dan Fletcher's lab to work on cell mechanics and the biophysics of the cytoskeleton versus Rebecca Hill's lab and thinking about chromosome biology. And I really had, great experiences in both. I ended up going into the Fletcher lab partly because one of my undergraduate mentors was also there. And so I felt like, I wanted to continue that mentorship relationship with him. And I also got along with Dan really well. But I always kind of had a torch for chromosome biology and chromatin. And so when it came time to think about postdocs, and even before that, when I was choosing what classes to teach during graduate school, I I TA'd in chromosome classes as well. And so for choosing my postdoc, I decided that was sort of my last chance to go back to the chromatin world. I was really attracted by this idea that chromatin structures are closely related to the processing of genomic information. And I thought it would be a great opportunity to integrate all of my interests. And I found that actually that's really panned out. So I think the intuition that I built in Dan Fletcher's lab, thinking about the cytoskeleton's self-organization properties, thinking about these dynamic subcellular structures and how they come together, has really informed the way that I look at chromatin. But the fact that we use sequencing-based assays that I learned as a postdoc in Will Greenleaf's lab allows us to combine that structural information with the flow of information from genome sequence all the way through transcriptional outputs. So it's really, I think I found my happy place after a fairly long and winding journey.
Yeah, that's where we all want to be, right? So that's good for you. So when I understood this correctly, you were raised or are from the greater New York area. So
Well, sort
yeah,
of.
go ahead.
So I'm originally Romanian. So I
Okay.
grew up in Romania and Eastern Europe until I was I 9 or 10, and then immigrated to the suburbs of New York with my family.
Yeah, what I was getting at is that now you're at Rockefeller University. So do you wanted to end up there, or how did you get there?
Yeah, so I spent a very long time in the Bay Area, and when I was applying to faculty jobs, I mostly concentrated my applications between the Bay Area and New York, because my family is here, my partner's family is here, and so we thought it would make life easier. And we also, I really love New York. I love how diverse it is. I love being in a big city, and it was always kind of a dream to live in Manhattan and work at one of these big New York institutions that I really admired when I was a kid. And so I honestly never thought I would end up at Rockefeller. It's been a bit of a dream come true.
So let's come to your science that centers around probing chromatin architecture and developing new methods to do so. I want to start with a paper from 2015 out of Trends in Cellular Biology or Cell Biology. There you state, we discuss how the paired end paradigm can be used to interpret sequencing data as biophysical measurements of in vivo chromatin structure. So does that mean that this can be used in a way similar to HiC?
So I think the paired end information can give you an advantage over the older generation of methods that just looked at single end data sort of distributed over the genome in terms of their density, because you, depending on exactly what method you use for paired end sequencing, you can get some kind of information about spatial coupling between pairs of loci. So you mentioned HiC. That's a great example of a method that uses ligation of spatially proximal loci to read out the folding of chromatin at larger length scales. And we have now been working with and further developing more high-resolution methods like Micro-C, and we're also working on a new variant of proximity ligation with alternative fragmentation that we hope will give even further benefits. But basically, by cross-linking and ligating together nucleosomes, we can understand how they contact each other within the 3D space of the nucleus. Now, I've also developed an alternative method that I'm happy to talk about called RICC-seq, in which it's ligation-free and we look at spatially correlated cleavage. But even if you don't do that, looking at one-dimensional methods like a TAC-seq that effectively just digest the genome and look at the digestion products can give you important information about nucleosome footprints or the footprints of other DNA-bound proteins if you can extract out the paired end information, so where the two ends of each fragment are.
But if you do, and sorry to go into the details here, but if you do ATAC-seq, for example, what you get is like only one linear piece of DNA, right? You don't get like two pieces that are fragmented together. How would that then yield like a 3D information?
Not 3D, but one-dimensional information about, for example, the protection of DNA by the nucleosome. Yeah. And so my lab is really focused on the mesoscale of chromatin organization, how chromatin behaves on the scale of one or a few kilobases of DNA, and the relationships between wrapping of DNA around a nucleosome, the spacing of nucleosomes, and their three-dimensional interactions are very intimate, and you can't really disentangle them completely. And so I think, we think about printing, we think about spacing, and we think about 3D interactions together.
Okay. So you already touched upon my next question, because that would have been about RICC-seq, because this was published in Nature, and based on my research, this was the next publication on the time scale. Could you talk about RICC-seq and how this works?
Of course. So when I started my postdoc going into the chromatin world, I was really interested in mechanisms of silencing and how heterochromatin maintains the DNA that it occupies in a silenced state. And the fundamental question that I wanted to address was whether heterochromatin is one thing or many things. Is there structural diversity? Is there a relationship between the biochemical factors that give rise to silencing, such as HP1 and the polycomb repressive complex? And I should say, it's more like it's correlated with. Give rise is perhaps too strong a word. But I was really interested in the nature of heterochromatin and how its 3D conformation can perhaps give mechanistic insight into how silencing works. And at the time, there really weren't a lot of methods to probe how DNA and how heterochromatin are folded in situ in cells. There was a lot of work, of course, from in vitro reconstitution, and there was this model that DNA can, and chromatin, so DNA folded into chromatin can adopt a beads on a string conformation in which the nucleosomes are stretched out and the linker DNA is very accessible, versus a 30 nanometer fiber conformation in which the nucleosomes stack with each other in a tight and very structurally conserved way with a lot of order, such that the linker DNA is buried in the center of this effectively solenoid or cylinder of DNA and is inaccessible.
So can't be digested by many enzymes.
Right. So cannot is refractory to enzymatic digestion. And when people started to work in cells and started to use methods like X-ray scattering and electron microscopy, groups like Kazuma Eshima with Mikhail Eltsov and David Bassett-Jones did not see evidence of this quote-unquote 30 nanometer fiber that had been observed in vitro inside your typical cell. And so this model of the 30 nanometer fiber that was in all the textbooks and was thought to very simply explain repression and compaction in heterochromatin was really called into question around that time. And so we wanted to ask, okay, if you probe in a cell in its native context where you have a lot more dynamics, a lot more heterogeneity, and all sorts of physical effects like crowding, and at the time we weren't thinking about phase separation, but of course phase separation plays a role too. What does chromatin actually look like? And so we were looking at probes that could get into heterochromatin because most of the enzymatic assays at the time, so this was in the Greenleaf lab where ATAC-seq was developed. And when I started, ATAC-seq was not a thing yet. DNA-seq was primarily the method that was employed to look at accessible chromatin. But all of those were very good at probing the open parts of the genome. And so what about heterochromatin? Well, upon looking at ideas for chemical cross-linkers and other things that might give us 3D information about chromatin folding in cells, we came across some work from the 1990s from the radiation biology field by Björn Rydberg, who had looked at the DNA fragments that come out of irradiated cells that were exposed to ionizing radiation of different types. And what Rydberg found is that the profile of DNA fragment lengths actually has very well-defined peaks that have lengths corresponding to known features of chromatin, such as the subnucleosomal helical gyre size, so effectively one wrap of DNA around the nucleosome core, single mononucleosome units, and then higher order contacts and higher order peaks in the fragment length distribution that were consistent, at least, with what you would expect if nucleosomes stacked with each other in an alternating fashion. So if nucleosome N stacked with N+2 in a zigzag sort of tetranucleosome unit, then you would have very close contacts within the gyres of nucleosomes N and N+2. And if those contacts were cleaved at the same time in a spatially correlated way, then the fragments that come out would have those lengths. So this got us very excited because radiation can penetrate cells no matter what the chromatin state. You don't have to have permeabilization, you don't have to have fixation, and you don't have to have sort of enzymatic digestion and diffusion of enzymes into the chromatin. And so I set out to take these experiments that had been done prior to the sequencing era and adapt them and adapt this idea to the generation of a method that could be then put onto an Illumina sequencer so that we could read out what is this fragment distribution at base pair resolution and also develop epigenetic state-specific fragment length distributions. So we could ask, how does the chromatin fold in euchromatin versus heterochromatin versus different types of heterochromatin?
So the radiation would enable you to look at heterochromatin only because it would completely destroy the euchromatin?
No. So we actually get signal everywhere. So unlike ATAC-seq or other open chromatin focused methods where the enzyme primarily just goes to open chromatin and that's what you get, RICC-seq gives you fairly even coverage across the genome. It is still slightly enriched for euchromatin reads, but it's something like 15, 20% at most, not 90%.
Could you maybe quickly, because you walked us through the method now, summarize the advantages of this method in one or two sentences?
Yeah. So the advantages of RICC-seq are that you can probe cells without having to cross-link them. So you don't have to worry about artifacts that might be induced by cross-linking. And furthermore, because it's non-enzymatic, it's technologically orthogonal to a lot of the enzymatic methods that are typically used in the field for both 1D and 3D chromatin mapping. So for example, enzymes like DNase 1 and micrococcal nuclease or MNase are well known to have sequence biases and where they like to digest. And then for us, who are also thinking about the 3D aspect, we have to think not just about sequence biases, but also accessibility biases, because we're not trying to just probe enzyme accessibility, we're trying to probe the 3D contacts. And so the nice thing about radiation is that because it's a physical process, you don't have to worry about the enzymes cleaving the genome with a bias. Now, of course, as a method development person, I will readily acknowledge that you cannot completely eliminate bias from your protocol. But for us, the biases come further down in the library preparation, and they're a bit easier to control for in that way because we always run, for example, genomic DNA controls where instead of irradiating chromatin, we're irradiating purified DNA from the same cell and running it through the exact same protocol to account for the bias. So what this has allowed us to do gets into some work that that my lab is working on right now and hopefully finishing up soon, is to cross-validate between independent technologies so that we can be confident that the answer we get and the conclusions that we make are as close to truth as possible. So one of the things that we're interested in is how the short range, the kilobase scale of chromatin is regulated by linker histones. And the problem with working in this field of mesoscale chromatin organization is that there is no kind of known gold standard. We don't have very good positive controls where we know exactly what it looks like, because that kind of thing can be done in vitro, but you don't know if in vitro and in vivo correspond very well because of all of these crowding and heterogeneity and dynamics effects that we talked about. So rather than having a sort of standard candle that we can compare against, the best that we can do right now is probe what the structure looks like inside cells with two completely independent methods, and we're using Micro-C and RICC-seq. And then if we get the same answer, then we feel like we can trust it because each of them have their limitations, each of them have their biases. But because the processes are completely different, we feel like those should, not cancel out, but they should compensate for each other.
How good is the correlation usually with those two methods?
We are getting, so when we estimate what the, for example, things that are relatively easy to measure, like the spacing between nucleosomes. So that's one of the pieces of information that we can extract from RICC-seq, from Micro-C, and then from other methods like ATAC-seq. We've seen that there's good agreement across the methods that way. So we can get it within a few base pairs. And so that gives us confidence that, it's working pretty well. And when we look at changes in the fragment size distributions, which effectively are, our readouts from the different methods, or in the case of Micro-C, it would be contact distance distributions, the trends, and the way that the contacts change and the directions in which the peaks shift are all self-consistent when you perturb linker histone.
So I want to come to a slightly different path of your career and then come back to the RICC-seq again. So we're taking a different road. So this was or is still on bioRxiv, so as a preprint, and there you present an open source protocol for the generation of multi-purpose hyperactive TN5 transposase, including its benchmarking, cut and tag, and bulk single-cell ATAC-seq. So can you talk about this effort and why you are making your own transposase?
So this project was actually the brainchild of my grad student, Jan Soroczynski, who has been working on enzyme optimization and enzyme generation on multiple fronts within the lab. And when we tried to generate our own transposase, both in this lab and in the past, in the Greenleaf lab, we found that it's actually very hard to get stable enzyme that retains activity in storage for more than a week or two at the scale of production that is feasible and reasonable for a lab that's sort of making its enzyme in-house. So not a big core facility or company that's producing huge amounts. And Jan was learning how to do protein purification and working with Dina Oren at our structural biology core facility. And so he tried to see what would happen if you just make protein G TN5 fusion rather than just TN5, with the idea that the protein G might help its stability and solubility. And he has biochemistry training from his undergrad as well. And he's very careful about optimizing culture conditions, purification conditions, and buffers. And so he found that actually making the protein GTN5 and then applying it to not just cut and tag, which is sort of the initial application that we had, but also just regular ATAC-seq, when we benchmarked it, we found that it behaves very well. And this is a project that was sort of simmering along for a while. And much to our delight, it turned out that the enzyme was stable in storage for more than a year and a half. And so when we found that, we thought, this is a great resource for the field because it's not necessarily obvious in a lot of publications, but personally, from talking to people trying to make it and, talking to people at conferences, we knew that this was a challenge in the field, that your average lab that wanted to make TN5 for development of novel methods was really having trouble generating active enzyme. And so we hope that this Open TN5 project, as we're calling it, will sort of make it much more accessible to develop novel epigenetic methods using TN5 and protein GTN5, because it makes it, easier and more affordable for a broader variety of labs to just make their own enzyme, load it with whatever adapters they need, and try out new protocols.
So another paper that is out in bioRxiv, and that was coincidentally uploaded in the same month, so July 2024, follows up on the RICC-seq paper. There you developed a flexible modeling and simulation framework to enable the interpretation of RICC-seq data in terms of oligonucleosome structure ensembles. Can you talk about this study and what you found?
So, this was a collaboration with Andy Spakowitz from Stanford that actually started from our discussions back when I was still a postdoc, because with a new assay like RICC-seq, we need to build up the computational infrastructure and the data analysis infrastructure to make sense of the data. And we wanted to go beyond just qualitative observation and interpretation of the RICC-seq fragment length distributions to start making quantitative predictions about what are the properties of chromatin and chromatin fibers that are being assayed with a RICC-seq experiment. Now, the challenge that we faced in doing this is that RICC-seq, by necessity, is a bulk experiment. It's not single molecule, because to get enough DNA fragments to build up a histogram of fragment sizes, you can't just get one piece of a chromosome. It's possible to do things like single cell high-C because you're looking at much larger structures. But when you get down to the scale of nucleosomes, you cannot simply extract out enough DNA fragments from a single cell to build up the data analysis that you need. So we're dealing with bulk. We're dealing with a population of cells and a population of chromatin structures that we know from the emerging data that they're heterogeneous. We know that there's dynamics and heterogeneity in chromatin. In some ways, it behaves somewhat like a liquid. And so how do we deal with that? Our solution was to use simulations to build up ensembles of chromatin conformations that are consistent with a single set of basic nucleosome parameters, such as DNA wrapping around the nucleosome, spacing of nucleosomes, and an effective interaction potential between nucleosomes that causes them to want to be attracted to each other and to want to compact into smaller clusters. And we're not doing things that are as detailed as, say, the work of Tamar Schlick or Rosanna Colapardo, where they're really building up from atomistic simulations all the way to coarse-grained representations, and including all of these other factors like electrostatics. We just wanted a basic representation of what the ensemble might look like based on the geometric properties of chromatin. So we did that thanks to some really beautiful work by Nicole Pagani, who was a research assistant in my lab. And then the project was taken over by Ariana Brenner-Clerkin, who used this framework, did a lot more validation on it to make sure that it looked that our basic parameters were in line with the literature, and then used these ensembles of chromatin conformations and the predicted RICC-seq fragment length distributions from each ensemble to train a neural network that we could then apply to experimental data. So right now we're in the process of revising this work and tweaking the neural network parameters so that we can apply it to as broad a range of data sets as possible. And we hope that this will be a useful tool to allow us to extract out parameters like nucleosome wrapping and internucleosome interactions, or at least effective interactions, that are very difficult to measure in situ. And for validation, we use things like the nucleosome spacing, which we can also extract from this data, but is also measurable through independent means.
And this again loops, I guess, very well back to your interest in physics and biophysics, right? Because this is right at the crossroads of all those things, right?
Exactly. Yeah. So I'm very much a biophysicist at heart, and one of the things that I always bring to a problem, and I try to instill in my students as well, is trusting your physical intuition and not being satisfied with an amorphous cartoon of how things work, but rather trying to understand it in terms of basic molecular principles and physical principles of how polymers behave, how diffusing molecules behave, and how biochemical interactions work.
So on your website, you list some areas of your research. Maybe we can now talk about what you're planning, maybe, let's say, in the next future, so the next 5 years. For a bit, the first bullet point on your website is probing short-range chromatin folding. And this probably extends on your RICC-seq work, right? And your RICC-seq development. So what are you planning in this area?
Yes. So we have some exciting work that is in process right now and hopefully finished soon to understand how linker histones regulate the short-range folding of chromatin. That is as I mentioned, combining RICC-seq with Micro-C so that we can, make the strongest conclusions possible. And hopefully this can also give us insights into how structural proteins like the family of linker histones that are expressed in most human cells regulate the overall transcriptional landscape and the overall physical landscape that is seen by a diffusing transcription factor or by other chromatin interacting proteins. These proteins, like linker histones and other architectural elements of chromatin, are really fascinating because they seem to bind very broadly, so they're not necessarily having locus-specific effects. And yet, they're very important in regulating things like cellular plasticity and the overall, I say chromatin architecture. That's the name of my lab, because that encompasses how chromatin genome-wide responds to stimuli and allows biological processes like transcriptional initiation, DNA replication, and DNA repair to happen.
So this feeds probably also into the next bullet point, because there you say molecular regulators of chromatin architecture, and on this below this bullet point you list factors like linker histone H1 and macro H2A. You talked about it yet, but this is basically the same thing, right?
Mm-hmm. So macro H2A is another project we have going on in the lab. It's we tend to go after these black sheep of the epigenetics family, where they bind very broadly. They can be very challenging to study, but we think that they're very important in shaping how chromatin behaves. So macro H2A is also one of these proteins that tends to be expressed more in differentiated cells. And although there are some examples where it seems to activate transcription, it's largely a repressive factor. But it has been very challenging to understand what its mechanism for regulating transcription is. There are many examples showing that it plays a role in cancer as a tumor suppressor, depending on the context. But we're trying to see what effect it has on the chromatin fiber organization, both at the nucleosome and oligonucleosome scale, as well as at much larger length scales of compartmental organization between active chromatin and heterochromatin.
And the last bullet point on the list is chromatin organization, cell cycle exit, and robustness of the epigenome. So I guess this is then more the translational part of your work.
Yeah, so this actually gets at something that I learned mostly during my postdoc, where I wanted to interact as much as possible with colleagues in other labs nearby, and I was very lucky to be next door to Monty Winslow's cancer lab. And so I kind of made friends and started talking to Xin-Heng Chiu, who was one of the Winslow lab postdocs. And I found that we approach science from very different perspectives. He was a hardcore cancer biologist, and I was, a very bottom-up sort of molecular thinker coming from biophysics. And I realized that a lot of the questions that they were thinking about could potentially have help from biophysics, but it would have never occurred to me to work on that if I wasn't talking to them. And so when I was starting my own lab, I wanted to look at what areas have a lot of open questions and are closely related to these topics of chromatin silencing and epigenome rearrangement that I was very excited about. And it looked like senescence was a field that was relatively understudied. It had very fascinating chromatin phenomena, such as formation of these heterochromatin foci that appear in senescent cells, and massive rearrangements of the transcriptome, such that whole transcriptional programs get activated when cells transition from cycling to quiescence to senescence. And so I thought this was a really exciting area where we could collaborate with people in the tri-institutional environment here at Rockefeller, because we're literally just across the street from Weill Cornell and from Sloan Kettering Cancer Center. And so when I started here, I initiated a collaboration with Andrew Koff at MSKCC to look at chromatin dynamics during senescence induction by cancer therapy, by cell cycle arresting cancer therapy. And this project then led to a related project in breast cancer. And now we're writing up the liposarcoma project with Andy, and there's a couple of papers that are in the works related to that. And we also have a really exciting new direction in breast cancer where our basic curiosity about the dynamics of chromatin accessibility and transcriptional regulation led to the discovery of a potential marker gene for cells that can resist therapy, or at least tolerate therapy. So we're actually learning a lot right now as my postdoc, Justin Rendelman, is kicking off collaborations with the therapeutic development folks here at Rockefeller and MSKCC and Cornell. And we're thinking about actually making an antibody to target it. So we're, I never thought I would get to this kind of research, but it's really fun where curiosity takes you. And I think the questions that emerge out of this more translational work are now feeding back to what we think about in terms of 3D genome organization.
So for the last 40 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
One thing I'd like to mention is a very exciting collaboration that we started. In addition to the collaboration that I mentioned in the cancer world, we worked with Yael David at Sloan Kettering and Rob Schwartz at Weill Cornell on a project to understand how chromatin assembly regulates transcription on the hepatitis B virus genome. And this was a very exciting and fruitful project because it let us use our chromatin expertise in a new field in virology. It let us combine our work assaying chromatin and transcription in cells with work on in vitro reconstituted substrates made by the David Lab. And it also led to the discovery of a potential novel therapy for HBV that we're very excited about. So this just came out online in Cell, and it's an area that my lab, as well as our collaborators, are excited to keep pursuing in follow-up work, including thinking about how the silencing of the HBV genome is accomplished by the host genome, and looking at 3D chromatin organization and its role in that process, as well as going in a more single molecule direction to look at heterogeneity between viral genomes and the same cell population. And that's fun because it's sort of the next step beyond what you can do with the paired end technologies that we talked about at the beginning, where you can look at correlations between cleavage events on either side of, say, a nucleosome in a footprinting experiment. But now you can actually look at correlations over a longer length scale with longer read sequencing and methylation footprinting. So it's an exciting area. It sort of marries very well with our interests in the basic biology of chromatin, and it's one of the directions that we're thinking of going forward into in the future.
So the treatment you were mentioning, is that like still in the basic research phase, or is this already transitioning to clinical trials?
It's still in the basic research phase. So we identified a destabilizing compound that compromises nucleosome occupancy on the hepatitis B virus genome. And this compound is part of the curaxin family of small molecules. It's currently in clinical trials for some cancers in a somewhat different context because it affects the overall activity of the FACT complex. But because it already is in clinical trials for a different purpose, we're excited to see if it could potentially be a promising therapy for HBV as well. Right now, we've done in vitro cell line work on primary cells as well as cell lines, and that looks very good. So it seems to be effective at disrupting the viral life cycle in the nanomolar concentration range, but we haven't yet taken it into mice or clinical trials.
So my last question is, what is your definition of epigenetics? So your personal definition.
My personal definition of epigenetics. I remember when there was a lot of debate about whether epigenetics is purely cross-generational, and so usually the word that I end up using is epigenomics rather than epigenetics, so that we can really be focused on the structural and biochemical mechanisms that act on DNA sequence to regulate how information flows from the genome to the functional space of cellular operations, of transcription, of translation, and so forth. So I think, to me, epigenetics or epigenomics are really about how does a cell work as a computer, right? What's the hardware of the cell? And that very much gets to my initial fascination back in high school of how do cells process information, and can you make a computer out of biological materials? Well, evolution already has, and epigenetics is the key hardware that makes it happen. So it's a very exciting place to be as a scientist.
Yeah. So thank you, Viviana, for your time and for being on the show.
Thank you. It was a pleasure.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.